Okay. Hi, everyone, and welcome to today's lecture, which is part of the Psychiatry Special Lecture Series. And I'm delighted to welcome Professor Christian Tamnes from the Department of Psychology at the University of Oslo. And Christian's research focuses on structural and functional brain maturation in childhood and adolescence, and how this neurodevelopment relates to mental health, cognitive processes, and socio-environmental factors. And with the advent of large-scale longitudinal neuroimaging, we're really just beginning to map out typical brain development. The kind of overall goal being to create a growth chart of brain development so that we can identify deviations from this typical development at the earliest stage possible, and then kind of, personalized interventions accordingly. And Christian's been involved in some of the first longitudinal neuroimaging studies in adolescence with multiple time points. And this has allowed Christian and colleagues to examine things like individual variability in brain changes across adolescence, which haven't been possible with cross-sectional data to date. So today, Christian will give a talk on how we can study brain development in childhood and adolescence using MRI signal intensities. And he'll first introduce his research group in Oslo and some of the relevant quantitative MRI measures, and before presenting some studies from his group on age-related differences in brain microstructure and its associations with cognition, mental health, and genetic factors. There's a Q&A box, so please feel free to pop your questions in there throughout the talk, and then we'll have time for questions at the end. But without further ado, I'll hand it over to Christian to kick us off.
Thank you very much, and thanks for the introduction and for having me. I wish I could come visit. I've never been in Edinburgh, actually, so that would have been much nicer, but it's also nice to meet you here. So yeah, just please interrupt me during the talk if there are questions or if anything's unclear. But I will, let's see. So I'm gonna say just a few words about my group and what we are doing, like on a very overarching level first. And then I've chosen a quite specific topic, I would say, for what I'm gonna talk about, namely how we can use MRI signal intensity measures, which are less used measures, I would say, to look at brain structure and structural development in childhood and adolescence, which are the kind of periods I'm most interested in, and how this brain development then relates to cognitive development and psychopathology. So hopefully this will be of interest. Yeah, just a few words about my research group first. We're a small research group at the psychology department at the University of Oslo. The group is called Neurocognitive Development. So we have a small group of postdocs and PhDs and master students and so on. And we are also so lucky to have both Marike Boos from Leiden and Kate Mills from Oregon in kind of part-time positions with us. So that's a very good international addition to the group, I would say. And very generally, we are interested in how brain development and cognitive development is related to mental health and well-being. And as I said, adolescence is kind of my main interest. And we're also interested in kind of all the type of predictors for this development. So we're interested in genetic factors, socioeconomic differences, and also very much environmental and psychosocial factors that can kind of affect our neurocognitive development and ultimately lead to increased risk or resilience. And we've also done some work on whether we can, on trying to better understand sex differences in the brain and in brain development. And ultimately, we want to see if we can kind of use these approaches to better understand sex differences in the prevalence, clinical presentation, and course of mental health disorders. So this is like very, on a very overarching level. And in terms of methods, I think what I have been working most on is structural MRI. morphometry, but also diffusion imaging. And what I'm going to talk about today is also structural imaging. But I would also like to say that we're now, in recent years, we've increasingly focused on EEG also, and we're also doing more, starting up some fMRI projects. So we're kind of expanding out a bit in terms of methods. All right, so the research group I presented is part of two different research centers at the university. So the first one is a center called Promenta, which we got the funding for in 2018, and we started in 2019. So Promenta is the abbreviation, and the center is called the Research Center for Developmental Processes and Gradients in Mental Health. And this is an interdisciplinary center spanning from genetics, neuroscience, psychology, sociology, and intervention research. And the kind of overarching aims here is to, at all these different levels, look at developmental processes and also social differences in mental health. And we're mainly here interested in kind of the full range of variation and in common mental disorders. We're also affiliated with a larger center called Nordment, the Norwegian Center for Mental Disorders Research. And this is a very big center with a main focus on severe mental disorders and kind of a strong foundation in molecular genetics and imaging, I would say. Yeah, so that was, I think that's enough of a presentation, and we can move on to the science. So as I said, I'm going to talk about some studies we've done on whether or how we can use MRI signal intensities to study brain development in childhood and adolescence. So I'm going to start off by presenting these measures and tell you a bit about what they are, because they might be new to some of you. And then I'm going to present some studies we've done using these to look at kind of neurocognitive development. And this work is mainly based on the PhD work of Linn Nordbom. So the credit for this goes to her. She's now a postdoc in my group, and we're still kind of venturing into using these methods in the ABCD study and so on. So we can talk about this towards the end if we have time. All right, so what I'm going to talk about is then some kind of less explored or less used MRI measures to study brain structure. So we can start off by looking at the usual suspects, the most kind of standard brain structure measures that most people use. And these can be kind of divided into volume-based measures or surface-based measures that look at different morphometric aspects of the cerebral cortex. And these surface-based measures, the usual suspects here are the thickness of the cortex and the area, and the kind of combination of these two is cortical volume. Now, importantly, both these kind of volume-based measures and the surface-based measures are in part based on local variation in signal intensities. That is the shades of gray in the voxels at different places in the brain. And in the T1 weighted structural scans, these signal intensities range from black in the ventricles, and we have kind of gray intensities in the subcortical structures and the cortex, and we have quite bright intensities in white matter. So, yeah. We can say that both from FreeSurfer, which is one of the most common softwares we use to get these kind of usual suspect measures, these signal intensity values provide very important information when kind of calculating the usual measures. So this is a classic figure from Bruce Fischel from one of the methods papers on FreeSurfer, and we see that signal intensity values from different brain structures is one of the types of information that is used to segment the brain. So for example, the ventricles show very much lower intensity values than other subcortical structures. However, we also see that many of the intensity values overlap a lot. So for example, the hippocampus and amygdala have almost identical distributions of intensities. So in addition to the intensity values, FreeSurfer also uses spatial information. For example, we know that the amygdala is always anterior to the hippocampus, and this kind of information is built into the algorithms. But still, the intensity values is one of the types of information that is used to perform whole brain segmentation and to derive volume-based measures. When it comes to the surface-based measures, the intensity information is even more important. So here is kind of a close-up coronal picture of the brain. And we see that FreeSurfer defines the boundary between the white matter and the gray matter, which is here marked with a yellow line, and the outer boundary of the cortex, what is called the pile surface in red here. And the kind of definition of where these boundaries are is in large part based on changes in intensity values. So the starting question or the kind of premise for what I'm going to talk about is, why don't we just use the signal intensity values themselves and kind of study how these relate to age, to symptoms, to cognition, and so on? Because they are kind of critical information also to derive the more usual suspect measures. And the rationale or the reason for doing this would be that we would think that local or spatial variation, but also individual differences and age-related differences in signal intensities would kind of reflect biological differences in the brain. So we can come back to this a bit later. But in more general terms, if we take a step back, I think we're always looking for kind of new imaging measures to use, right? And when I think of this, I think of it as we're trying to either increase specificity or we're trying to increase sensitivity. And with regards to specificity, it's been proposed that these signal intensity measures and also something called contrast measures that I will talk about soon do provide more biological specificity than some of the other types of measures that we typically use, for example, cortical thickness. So it has been proposed, for example, that these intensity measures are quite sensitive to, or quite specific, sorry, to a degree of myelin content in the brain. With regards to sensitivity, I think we can think of this as looking for measures that explain additional variance in the phenotypes we're interested in. So the question here is whether these intensity values or intensity measures tell us something different than cortical thickness and cortical surface area and so on. So I'll come back to this sensitivity question towards the end when we talk about multimodal imaging and whether they kind of explain additional variance beyond the usual measures. All right, so as a concrete example of how these signal intensity measures can tell us something interesting, I want to show you a study we did a long time ago, back in 2010, led by Lars Vestly. So in this study, we extracted gray matter intensities from the cortex, and we also looked at cortical thickness. And we looked at this in a cross-sectional sample of more than 400 individuals from 8 to 85 years, so a lifespan sample. And here are just 4 kind of selected regions to show you the age-related differences. And if you first look at the red lines, these are cortical thickness. We see that for all of these regions, cortical thickness show a monotonic age-related decrease. So it's the cortex is getting thinner both during development and during adult age and in aging. And this is kind of a puzzle, because we think that somehow we think that the thinning we see during development is a positive thing, right? It reflects maybe specialization or increased efficiency in the cortex, while in aging we think it relates or reflects more a degenerative process. But the cortical thickness measure doesn't really differentiate much, right? While if we instead look at the black lines, the signal intensity within the cortex, we see that this measure does differentiate between developmental changes and aging related changes. So I think this is a nice example of that these kind of measures can pick up something different than the usual suspects we're using. One of the problems with the signal intensity measures is that they are not constant across participants. And this is because there are both kind of MRI session and participant-specific optimization procedures that the scanner uses. So the scanner will kind of give you different intensity values from even the same person at 2 different scans. So what we have to do is to normalize the values in some way. And what we did in this study back in 2010 was to normalize the intensity values within the cortex with respect to the intensities within the ventricles. And the idea, of course, would be to try to control for or correct for kind of inter-subject variation in this kind of intensity scaling differences. Some better options might be to directly contrast 2 different intensity measures of interest. And this is what I'm going to talk about today. So I'm going to present 2 such signal intensity contrast measures. The first one is the one called gray-white matter contrast. And what we can do here, the actual calculation varies a bit from study to study, but basically we can start off at the yellow line, start off at the kind of boundary between white matter and the cortex, and we can sample intensity values both going into the cortex, either at kind of predetermined distances or at percentage different distances. And we can do the same thing down into the white matter, and we can then calculate the ratio or the contrast between gray matter intensity and white matter intensity. So this gives us a very kind of local measure of how different the intensity is within the cortex and in the white matter right beneath. The other measure, which has been used quite a lot and was originally proposed by Matthew Glaser and David Van Essen, is called the T1 weighted, T2 weighted ratio. Originally, it was actually proposed or called myelin mapping. What you do here is that you have to have 2 different structural scans, both the T1 scan and the T2 scan. and you register these to each other, and you can calculate the ratio between the intensity at the same points in the cortex between these two different scans. So kind of the big difference is that this is a purely intracortical measure, while the gray-white matter contrast is the difference in the intensities in the cortex and in the white matter right beneath. All right. Yeah, please interrupt me if I'm not clear on explaining this. So going back to the issue of specificity, both of these contrast measures have been proposed to be sensitive to myelin content. And as I said, the T1 weighted, T2 weighted ratio was even called myelin mapping originally. And myelin is, of course, I would say almost a holy grail when we're doing imaging. We're always kind of hoping that we get imaging measures that pick up myelin differences. And in terms of development, this is, of course, very interesting because we know that myelination is a very protracted process that keeps going throughout adolescence. And we also, of course, know that it increases not only speed of neural communication, but also frequency and kind of consistency in signaling. And also, just as a reminder, we often think of myelination as something that's predominantly in white matter, right? Around the axons in the major fiber bundles, but we also have myelination within the cortex. So this is, when we talk about the T1, T2 weighted ratio, we're talking about intracortical myelination, or possibly intracortical myelination, because more recent studies have, of course, shown, as has also been the case for other types of imaging measures, that the reality is probably not so simple. So these measures are, it's probably incorrect to refer to them as myelin mapping and so on, because they're not specific to myelin content. But either way, we do believe that they at least partly reflect myelin differences. And one reason we believe this is that the spatial kind of variation in the maps is very similar to the known regional variation in degree of myelination. So we know that the primary motor sensory regions are highly myelinated, and we also see that these kind of measures pick up on that. So, so here on the left, you see like an average gray-white matter contrast map. In this case, we flipped the intensities just to have it in the same direction as the T1, T2 weighted ratio map. In addition to that, we kind of see that they look similar to known regional variation in myelin content. We also see that these two maps are quite similar. So we see that the regional kind of variation in gray-white contrast and in T1, T2 is mostly quite similar. All right, so this was the background part. So I'm going to move on to present some studies where we have tried to use these measures to study brain development. So in the first study I'm going to present, we used data from the Philadelphia neurodevelopmental cohort, which I guess many of you know. It's an open access US data set. And there's questionnaire and cognitive data on more than 6,000 individuals in the ages 8 to 22. And then there's a subsample of more than 1,400 kids with imaging data. And I must say the beautiful thing about the PNC is that they're all from the same scanner. So there's no scanner variation. So what we did first, I must warn you, Lynn, who did this work, is a big fan of ICA analyses and so on. So there's lots of independent component analyses and principal component analyses going on. So what we did first here is to run an ICA on this gray-white matter contrast map. So we get lots of different components picking up local variation in gray-white matter contrast. And then we looked at the correlation between these different component loadings and age. So what we see here, it's maybe a bit small on your screen, but the first component had loadings across the whole cortex, and we see that this component had a negative association with age, meaning that gray-white matter contrast becomes more lower. So the difference in intensity within the cortex and in the white matter beneath becomes smaller. And we believe that one of the processes that underlie this is intracortical myelination, so that the cortex becomes brighter and more similar to the white matter, right? And on top of this first component, there's lots of regional variation that reflects faster and slower age-related changes. And these kind of components then have to be interpreted to be in addition to the general pattern seen in the first component. So notably, I also want to mention the study we did was a cross-sectional study, but very recently, the first longitudinal study on gray-white matter contrast came out from Sharif Karama's group at McGill University. And they also found kind of the same thing, that the gray-white matter contrast goes down with age during adolescence. So the next thing we did was to test for how individual differences in gray-white matter contrast was related to psychopathology. And for this, we used an analysis originally published by Dag Alnes in the group. So what Dag had done was to just line up all the questionnaire all the clinical items in the PNC dataset, and then run an ICA on this. So what we get from this is kind of data-driven clinical components. So we see, for example, in the first component really picked up on questions from relating to ADHD, while some other components picked up kind of different anxiety-related questions from different parts of the clinical interview. So we kind of then used these clinical components and looked at how they were associated with the gray-white matter contrast components. So this is what you see here. And what we found was that there were particularly, or specifically, the component related to anxiety and another component related to prodromal psychosis symptoms that showed regional associations with individual differences in gray-white matter contrast. And these are, so there were no associations for this kind of global component, the first gray-white contrast component, but there's some for the other components. And this is a bit tricky to interpret because as you see, the sign of them vary because they have to be interpreted together with the global component in the other components. But they do pick up at least some regional variation in this. And interestingly, the associations were in the opposite direction as some associations we saw with the general cognitive ability. This was the first study I wanted to present on gray-white matter contrast. And what we next did was to look at the T1 weighted, T2 weighted ratio measure. And for this, we had to use a different data set because the PNC does not have T2 weighted scans. So we had to use the PING study instead, another open access US data set. So the final sample we ended up with here that had both T1 scans and T2 scans from the same individuals and of good quality ended up being 600 kids. And they are also a bit younger. So the PING is quite unique. It starts from age 3 and goes up to 21. And a challenge here was also that the data are from many different scanners. So we had to control for scanner effects, which were very large, as they usually are. So what we did first was to then correlate the T1 weighted ratio, T1, T2 weighted ratio, and age. And we see a almost like global increase from early childhood to young adulthood. Actually, I did not include it here, but I think this was also, we found mainly a linear effect. So this kind of keeps increasing also across adolescence. And we see also that the effects are strongest in frontal regions for the most part. Surprisingly, what we did next was to look at associations between this contrast measure and general cognitive ability. And the surprising thing here was that the associations were negative. So the contrast goes up with age, but we find a negative association between this individual differences in this measure and cognitive ability. So here we're controlling for age in this analysis. So it's age-independent associations. So this is still puzzling. There are a couple of other small studies that find the same thing in development, but we actually don't have a good explanation yet. And so I think this needs replication, and we need to see if this is actually true. So I don't know, it might be that we're picking up on that excess myelination or too early myelination or something is a negative thing, or we might pick up on other biological processes. We don't know. All right, so moving on to the third and final paper I'm going to present, we're going to go back to the question of sensitivity. So what we wanted to do here was to combine these kind of contrast measures with the usual suspects when we do structural imaging. So for this, we combined the Philadelphia neurodevelopmental cohort and the PING data set. And then we had to look at, then we did not have the T1, T2 weighted ratio. So we're looking at gray-white matter contrast. And the final sample is 2,500 participants. So it's quite a large study. And we used a software called Flica, and this is part of the FSL suite. So this is an independent component analysis program where you can input many different types of imaging data at the same time, and then get multimodal components. So our input data for this analysis was vertex-wise cortical thickness, cortical surface area, and gray-white matter contrast. So we have kind of three maps that are spatially aligned to one another, and we get independent components that can have different loadings on these three different measures. Similar to what we had earlier, you get lots of different independent components from this. And I'm not going to show you kind of the full list, but I'm here showing you the first component, which is also the one that explained the most variance in the data. And we see that this component had very high and global loadings on gray-white matter contrast, and also some loadings from cortical thickness, but almost nothing from area. And we see that the component had a negative association with age. So this kind of indicates again that gray-white matter contrast decreases with increasing age, and also that this happens together with cortical thinning. And that in this age range, gray-white matter contrast is actually the measure that kind of picks up the most variance, which is interesting. And I think also this coupling between gray-white matter contrast becoming more similar and cortical thinning is interesting, because we do also speculate that, or we can speculate that this kind of observed cortical thinning we're seeing might be partly driven by kind of this myelination process going on at the boundary between the white and gray matter, which can affect also our kind of how we measure cortical thickness, right? So one more analysis we did in this study was to look at associations between these multimodal components and polygenic scores for neuroticism. So this is of course something we're interested in as a general risk factor that might predispose for psychopathology. But the results here showed only some very weak and bidirectional associations, which are not super convincing, I must say. So I think this was mainly like a null finding. And I think this is, in hindsight, not very surprising. I think these kind of polygenic scores, at least for things like neuroticism, that in themselves explain very little of the variation, of the phenotypic variation, we cannot really expect any strong associations with brain structure for these type of measures at the moment. But I'm happy to hear other views on this from people with more experience. So I'm quite new to this kind of analysis, but we are looking into it more now using bigger data sets like the ABCD study, so where we can get some more power, but still, I think it's very realistic to expect very small effect sizes. So yeah, I just want to mention we've recently also written a review paper where we go through a lot of this in more detail, also with contributions from Thomas Pause's group, including Nadine Parker. So check this one out if this is of interest. And finally, I'd like to thank Lynn and all the co-authors for doing this work, and also the providers of the data sets we used, the PNC and the PING study, and our funders. And I would also, this is my last slide, like to make a small advertisement for the virtual Flux conference. So I'm a part of the board for Flux Society. So this is the Developmental Cognitive Neuroscience Society. So if you're interested in this field, I would recommend this conference. So thank you.
Great. Thanks very much, Christian, for a really interesting talk. I think we'll just kick off with some questions. Does anyone have any questions for Christian about the presentation or about the work more generally? Heather?
So yeah, I have a question just to dip in before anybody else has a chance. So yeah, so many questions. That was a really great talk. Thanks very much. And these are kind of quite new measures, I guess, that aren't standard, as you say, and they're really interesting. And so one of the questions I had was how, in terms of your kind of developmental studies, looking at how they change over time, do you think these measures are explaining different variants to kind of standard measures like brain age and things like that? Do you think they're kind of explaining something on top of that, different biological processes, perhaps?
Yeah, I think they do pick up on, I mean, I didn't show you all the different components from this multimodal analysis, but they do. This probably depends on the age range we're looking at. So it might look very different in adults or in old age or something, but in developmental samples, this kind of link between this gray-white matter contrast and cortical thickness was was stronger than the link with surface area. But
yeah.
But I just found it at least interesting that the gray-white matter contrast was the measure that kind of got priority in this data-driven analysis and picked up the most variance to begin with. And I think it does provide some clues about, as I talked about in the beginning, where this cortical thickness measure is also like something which is very much influenced by this intensity values. So yeah, I'm not sure what I'm answering, but
no, that makes sense. Maybe it would be something nice to test in terms of developmental brain age versus these measures.
Sure.
The other question I had, if I can be cheeky, Neve, is have you looked at studies where you've kind of looked at early fixed life exposure? So, you know, early life stress and the effects on these kinds of measures?
To my knowledge, we've not done so. We have some small plans for it, but I think there are almost no studies. So these are very much underutilized measures, I think. In like at least the gray-white matter contrast measure is quite easily available when we have T1 scans.
Yeah.
So I think, and I would strongly suspect that they do pick up on variation which is different or on top of other measures. So that would certainly be one way to go, to look at, try to relate it to early life factors and so on.
Super, thank you.
But as I said, I'm not aware of any of these studies yet. So it's quite striking that, for example, the first longitudinal study on gray-white matter contrast was published like this month.
Yeah, it's so interesting. I feel like, I don't know, it's definitely not something you read about. Like, it's not really kind of considered in the you know, narrative about, you know, the kind of imaging, like psychopathology related imaging features. So really, like, I think, you know, because we're only explaining a small amount of variance, I think expanding our toolkit to kind of be able to probe the associations a bit more is really cool. Does anyone have any other questions? I have one while people are kind of forming their thoughts. So I was wondering, have they kind of, you know, like given the kind of, you know, like the kind of, you know, enigma studies and like the really big longitudinal and like cross-sectional studies in adults, have they looked at these in enigma? And kind of whether or not these like the gray-white matter contrast, you know, how does that relate like I know you touched on it briefly on how it relates to kind of maybe features of psychopathology, but has there, you know, how does that then compare to the kind of, you know, psychopathology related imaging features that we see, like, so like, you know, reduced, like, yeah, kind of like decreased cortical thickness and kind of volumetric reductions in the kind of hippocampus and stuff? Like, have there been studies comparing kind of these traditional methods with the kind of new intensity measures?
I'm not sure. I think perhaps Heather knows Enigma better than me, I suspect. But for developmental studies, which have been my focus, there's been like very, very few studies. So I'm sure there's some Enigma plans looking into this, but they do. Yeah, they do require you to go a bit deeper into than the kind of standard output measures, but it's not that hard either. So
yeah.
I don't know. Do you have some input, Heather, on this?
I'm not aware of anybody doing that within Enigma. I think, Niamh, you were particularly talking about comparing these kinds of measures to standard measures. I'm not aware of anybody doing that. I wonder if I might ask another question if there's nobody else. So in terms of practicalities, and this kind of links into the Enigma work as well, I suppose, is this, so that in terms of kind of the practicalities of deriving this is voxel-wise, presumably, data rather than, so you have to work from raw data to derive these measures, is that right? And is there software to do that, or you know, how does it work?
Yeah, so I think we've been kind of working from raw data. So let me just go back to slide here. But I just actually recently became aware that I think there are standard outputs from FreeSurfer now also with for gray-white matter contrast. So, but I should know this, but I think I just recently became aware of it, and then there's been some variation in how people have calculated this, so it's a bit different than what we did. So the figure on the right here is a paper by Kjetil Jørgensen on adult psychosis patients, which might be of interest to you. So, and for this study, I know that they did test out different kind of sampling strategies, and they found that it did vary quite a bit. So what they ended up doing, and this is kind of the method we adopted also, was to sample gray-white matter or gray intensity from many different steps and take an average so that it's not like super sensitive to whether you go so far or so far in, and the same for white matter. So I think if you want to do kind of these things, it's which would be what we would recommend, I think, is then you have to work from the raw data. But but I do think the kind of standard measure, there is an included measure in the standard FreeSurfer pipeline now, I think, which would be much more easy to use for something like Enigma. So so for Enigma, I would go with that one.
Yeah, definitely.
But I'm not, we haven't used it ourselves, but I don't know the details of it, but it's slightly different, but probably very highly correlated. Yeah.
Yeah. Thank you.
I can check into it and send you a link about it if you're interested.
Yeah, that would be super.
Yeah.
Yeah, I just wondering, while we have a few minutes left, and I just wonder, you mentioned briefly that you're kind of have plans to expand this work using the ABCD data set, and I just wondered, like, we work quite a lot with the ABCD study in our group, so if you could share your thoughts on our plans for what you're hoping to look at.
Well, we've not come very far, but I think it would be going even more into, because just utilizing this very big data set and looking at also as of the baseline data where you don't have the age variation, I think it would be super interesting to there are a couple of things I'm very interested in, and that's first of all, this kind of surprising association with cognition, which was strange. It's in the opposite direction that what we would expect. So it would be nice to look at this in a much larger sample to see if this is actually true. Or if it's driven by some kind of how we control for age or what, what's going on. And I think, so this is one avenue, and another is also kind of a polygenic score approach where you can then leverage the much larger data set. So in this combined study here, we had, it was large for the age-related analysis, but it was much smaller when we looked at the genetic data, right? So it's, so that would be more like, I mean, even very small effects might be of interest, but then you need a bigger data set to find them. So.
Yeah, well, ABCD is perfect for that. So, yeah.
But we are, yeah, it's a bit of a computing issue also when you want to go work from the raw data on these kind of things. So, yeah.
Great. Yeah, if no one has any other questions, I think we might wrap up. Heather, do you think that's?
Yeah.
Yeah. But yeah, I just want to thank Christian again for a really interesting talk. Definitely given a lot of kind of food for thought. So yeah, thank you very much, and look forward to seeing how your work develops.
Thank you for having me.
Great. Yeah, no worries.
Shall we catch up afterwards? Perhaps after Angel stopped recording and things?
Yeah.